The cell undergoes a sequence of changes, involves period of growth, DNA application, and cell division. This sequence of changes is called cell cycle. It comprises two phases: interphase, which is the period of non-apparent division, and the period of division is mitotic phase. Each phase is further subdivided into subphases. Interphase. The period between two consecutive divisions is called the interphase or resting phase. It is the period of great biochemical activities, and further divided into G1 phase, S phase, and G2 phase. G1, also called GAP1 phase, is the period of extensive metabolic activity in which cell grows in size. Specific enzymes are synthesized and DNA base units are accumulated for the DNA synthesis. Post-mitotic cell exit the cell cycle during G1 by entering G0 and remain for days, weeks, or in some cases for lifetime, for example nerve cells and cells of the eye. In S phase, the DNA is synthesized. Chromosome number is doubled. In G2 phase, the cell is prepared for division. For example, energy storage for chromosome movements, synthesis of mitosis-specific proteins, RNA subunits, and microtubule subunits. At each stage, there are specific check points which determine the fate of new phase. Duration of each phase is variable. In the case of human cell, average cell cycle is about 24 hours. Mitosis takes 30 minutes. G1, 9 hours. S phase, 10 hours. And G2, 4.5 hours. Whereas, full cycle in yeast cells is only 90 minutes. It is the type of cell division which ensures the same number of chromosomes in the daughter cells as that in the parent cells. Major steps of mitosis are similar in plants and animals. It takes place in haploid and in diploid cells. Mitosis is divided into two phases: karyokinesis, which involves the division of nucleus, and cytokinesis, which is the division of the whole cell. Karyokinesis. At the beginning, the partition of the centriole takes place. Two pair of centrioles separate. Each pair migrate to opposite sides of the nucleus, establishing the bipolarity. Three sets of microtubules originate from each pair of centrioles. One set, the astral microtubules, radiate outward and form aster. Other two sets are kinetochore and polar microtubule, compose the spindle. The kinetochore microtubules attach to chromosomes at kinetochores, and polar microtubules interact with polar microtubules from the opposite pole. These microtubules are composed of RNA and tubulin protein. Mitotic apparatus. Specialized microtubule structure. Aster and spindle is called mitotic apparatus. That attach, capture, aligning and separating the chromosomes, and ensure equal distribution of chromosomes. Division of karyokinesis. It divided into prophase, metaphase, anaphase, and telophase. Prophase. At start, the chromatin material gets condensed by folding and the chromosomes appear as thin threads. Chromosomes become more and more thick. Ultimately, each chromosome is visible, having two sister chromatids attached at centromere. Towards the end of prophase, nuclear envelope disappears. Nuclear material is released in the cytoplasm. Nucleoli disappear. Mitotic apparatus is organized. Cytoplasm becomes more viscous. Metaphase. Each metaphase chromosome is a duplicated structure which consists of two sister chromatids attached at a point called centromere or primary constriction. The centromere has special area, the kinetochore, with specific base arrangement, and special proteins where kinetochore fibers of mitotic apparatus attach. The kinetochore fibers of spindle attach to the kinetochore region specialized area in centromere of chromosome and align them at the equator of the spindle, forming equatorial plate or metaphase plate. Anaphase. It is the most critical phase of the mitosis, which ensures equal distribution of chromatids in the daughter cells. The kinetochore fibers of spindle contract towards their respective poles. At the same time, polar microtubules elongates, exert force, and sister chromatids are separated from centromere. As a result, half-sister chromatids travel towards each pole. Telophase, reaching of the chromosomes at opposite poles terminates anaphase and start. Telophase, the chromosomes decondense due to unfolding, ultimately disappear as chromatin. Mitotic apparatus disorganizes nuclear membrane and nucleoli reorganize, resulting two nuclei at two poles of the cell. Cytokinesis. During late telophase, the astral microtubules send signals to the equatorial region of the cell, where actin and myosin are activated, which form contractile ring, followed by cleavage furrow, which deepens towards the center of the cell, dividing the parent cell into two daughter cells.